Okay, with that, I'd like to introduce our next session. Dr. Robert Hoehndorf is an associate professor at KAUST and leader at the Bio-Ontology Research Group, also known as BORG. He holds a PhD from the University of Leipzig with prior experience at prestigious institutes such as Max Planck Institute and the University of Cambridge. Please join me in welcoming Professor Robert. Thank you.
Thank you very much. So it's my pleasure to be here. And thank you for the opportunity to present a little bit about what we're doing in the area of diseases, in particular rare diseases. So modeling diseases with AI is a very big field, so I have to focus a little bit. And I'll focus on rare diseases because they are a very high burden in this region of the world. And when we talk about rare diseases, the main question that we have is the diagnosis of a rare disease or supporting the diagnosis of a rare disease, because this can sometimes take 10, 15 years before somebody with a rare disease gets a diagnosis. So in general, what we have is, as input, we have some information about a patient, maybe gender, ethnic background, and so on, and then we have some phenotypes. Phenotypes are the signs and symptoms that we see, and for rare diseases, we very often also have a genotype. So we would identify DNA, germline DNA of this individual, and we have this given. And then the task is to find out why this individual has these phenotypes, signs and symptoms. So basically, we want to find usually a genomic variant, particular change in DNA of this individual that causes the signs and symptoms in this individual. Course here means if this individual would not have this variant, but would have some kind of reference DNA at this point, then they would not develop this phenotype. And the issue is that rare diseases is a challenge for machine learning in particular, and the reason is that we have very few samples. So this is the first reason. So we have very few samples by their very definition. Rare diseases are rare, so that we may only know 20 cases or 5 cases with such a disease. And there are many of them. So although each disease individually is rare, because there are maybe 7,000, 8,000, maybe more such rare diseases, only genetically based rare diseases, the overall burden is still quite high. So maybe 8% of a population suffers from a rare disease, although each is individually very rare. And the type of features that we get, because these diseases are very heterogeneous, we get different types of features. We have EHR data, we have imaging data, we have all kind of sensor data that we can have. And the search space is also vast, because our genome is about 3 billion base pairs long. Each of them, each nucleotide could be responsible for, or at least theoretically be responsible for this disease. So it's quite a challenge. And when we look at standard machine learning, so we could just learn, we take our data, we train some model, make some prediction. If we do this, then we want to make a prediction based on data, so we have some observation, we have some trained model, we make an observation. So if we take this, that obviously is not going to work. And it's not going to work because we have not enough data and too high dimensionality in order to make a reliable decision. And we could, of course, try to increase the amount of data, but because these are rare diseases, this does not work. So what I'm going to talk about is how we can maybe generate some internal representations first, reason about the state of this individual that comes with a rare disease. This might be considered a world model or some other kind of internal representation, but it's a model of this individual. And then we make a decision based on this. And we will, for this, as part of my talk, I will show how we can combine, or how we will combine, learning and reasoning, which is also the main area of my own research. So for rare diseases, we have to ask some questions. So the main reason why we want to have a world model, or one of the reasons, why we want to have this world model and basically not have this direct line from an observation to a decision, is that we can add a large amount of background knowledge. So we can constrain our search by how we know, for example, physiological processes in a human body happen. What connections are there in the human body? What anatomical structures are adjacent to each other and might exchange matter or fluids. So this information is not something that we would learn from an observation or from most observations. It's something that we would get from a textbook, for example, or from some database or knowledge base. And if we want to know which genetic variant causes a disease, we have to identify, we have to have a lot of extra knowledge that we would not get from information that we have about this patient. So for example, we need to first identify whether a variant actually affects the function of a gene. So whether it affects the function of a gene, we need to know structure, protein structure, and so on. Maybe may have to predict this. Need some biochemical properties, binding sites of this protein, and so on. And although this got a lot easier, it's not information that is about the patient, it's some basic biological information about human genes and how they fold. And it's a different type of information that we need. We also need to understand which variant is affected, or which gene is affected by a genetic variant. This works for some variants very easy, but if you imagine that there are variants that affect 1 million base pairs, so there might be 1 million base pair duplications or deletions in a human genome, then this becomes quite complex to identify which gene or which genes are actually affected by this variant. Or if you think of inversions. And so this is something that is only just now possible to investigate with long-read sequencing. And then we need some other information, so we need some interactions, so interactions in pathways, networks, and functions, so where are certain genes expressed, and then we get physiology. And so human physiology and anatomy. And the main issue, I don't want to go through this whole list, I'm not a clinician or a biologist myself, but the main thing that I want to highlight here is that none of this can be learned from the data that we have about our patient. This is something for which clinicians would study 10 years or longer, so that they develop this understanding of basic structures. And what we are working on are systems which can somehow utilize structured information, for example, about the human body and interactions in the human body, in order to make this decision, so that we can add this information to the data that we see about our patient or some patient and make a decision that combines prior knowledge and statements that would be entailed about it with data that we observe. So what we get is this kind of cycle in general that we work on. And on one side we have knowledge. This is usually structured and symbolic. It's something that is understandable to us. We can read it, we can communicate in this, and this is how, at least on a cognitive level, our thought processes help. So our cognition happens largely symbolically. On the other side, we have some data where we do pattern recognition. We need to identify some patterns in there, and I just call this learning in a very primitive way here. So this is a very high-level view. And there are two operations that we have between these two sides. And one is the embedding operation, where we take symbols and embed them in some kind of space where you can combine them with data, and then the opposite of this is the extraction information. And most of my talk will be about how if we design our embeddings in a suitable way, we get extraction for free. For biology or for health questions, often what we want to know is on the left-hand side, and what we are given, so what we observe is on the right-hand side. So we observe a genotype, it's largely unstructured, and we want to know what phenotype is resulting from this genotype. Or we see gene expression, we want to understand the function of a gene. So where is it expressed, what is the function, and so on. So let me talk a bit about the methods that we use here for the embedding before getting back to rare diseases and diagnosis. So basically, I take this definition of embedding. It's a very mathematical definition. It's different from what most machine learning researchers would use as an embedding. They would use a more relaxed version of this. This is a very strong definition of an embedding as a structure-preserving map. And it's how you would define an embedding maybe 20 or 30 years ago. Would be the main meaning of an embedding. And the reason so structure-preserving maps, there are many. So for example, if we take symbols and embed them in natural numbers, that's a structure-preserving map defined by Gödel, and it's called Gödelization. So it doesn't have to be in the context of machine learning. And the reason we define these embeddings is that the second structure may be more suitable for some operations than the first structure. And in our case, if we want to combine it with some modern machine learning algorithms, usually we want to perform some kind of optimization or apply gradient descent. So what we generally want to do is, for this form of knowledge-enhanced learning, is that we want to embed formalized knowledge bases, symbolic structures in Rn. And there are different approaches that we work on, or that my group works on. So some of them are graph-based or model-theoretic. I will just show you one approach today in some detail. But before I show this, I need to give a brief outline of the language that we use to express our knowledge. And this is a very, very lightweight language that I will focus on mostly today, which is called EL. It's a description logic. And it's basically standard syntax and semantics. So in computer science, this is basically first-year computer science. You get basic logic, and this is a small subset of first-order predicate logic, where you have some statements on the left-hand side. So this is how you can formulate statements, and then there's a semantics. Semantics is always given with respect to a domain, and you can define statements that are true or false using this. So this is basically the language that we deal with. And then for this language and some few extensions of this, we have worked on a number of different embedding methods over the years, starting in 2017, all the way to, I think we just submitted one last December. So I will focus on one, and this is Cat-E, which are based on category theory here, because they give us some properties that we can use later on, So basically what we start with some ontology or some kind of formalized knowledge base on the left-hand side, then we project this into a graph, then we use a graph embedding to get to Rn, and then we get the inverse relations. So we will do some link prediction or some knowledge graph completion methods. So this is link prediction in Rn. This gives us back a graph edge, or we can predict graph edges. And then we can invert this projection, where we take our symbolic structures, project them onto a graph, and if we can invert them, this is called axiom inference. And in the end, we get a statement that is true or false back. Now, when you look at research, there's a lot of research on the right-hand side of this. So there's a lot of knowledge graph embeddings, many, there are probably hundreds. For the right-hand side of how to get from a graph to Rn and then predict relations in Rn. What we focused on is the left-hand side. So how can we get statements that are true or false, not just basic facts as in a graph, but statements that are true or false, and get them into a graph. So this is this left-hand side, and there are a number of different methods. Some of which are by us, the first two, and the other ones have been used before. And I give you some example for this. So these graph projections, we have some axioms. So there are some axioms. You can see some example axioms or subclass axioms or equivalence class axioms. And we can represent them as triples. So this is the aim. We can get some kind of different kind of ways to convert them. So the first one is the L2vec or L2vec star. They use the same conversion for these axioms. And at the bottom, this is a domain-specific approach to do this. The big problem when you look at this is that these are not embeddings in my sense. And they are not embeddings for two reasons. They are not total. So they are not total because there will be, for example, negation or certain, so negation is not part of this, but negation is an operator that would be part of our language. And they are not injective or invertible. They're not invertible because You can see that quantifiers actually don't matter here. So quantifiers are treated, they will create the same triples independent of whether it's an existential or a universal quantifier. So what we are missing here is that these graph projections are not actually embeddings. We cannot invert them. And this means we cannot infer axioms. We cannot get statements back that we started out from. So we had statements that were true or false. Now we projected them into a graph, and we can't even get back from the graph. We can't get our statements back. So this is not great. And it also means it's not an embedding. So one thing that we tested on these embeddings, so a number of these, is whether they can compute entailments. This is a test, was also not developed by us, but we tried to test this, whether we can do this. We used here trans E in this. You can have a look at this. Basically, this is for gene ontology. It's a relatively large knowledge base that we tested whether we can perform entailment, so deductive entailment, using this method. And the answer is, when you look at this, largely no. So none of these methods really can compute entailed axioms. You can see this at the mean rank metrics that are like in the thousands. And if this is out of, even though this is out of 100,000 axioms, entailed axioms at this rank are not acceptable. So the question is whether we can do something better. And in particular, whether we can do something that actually is an embedding, or that would be an embedding in the sense that I have defined it. And for this, my student Fernando has worked on this. So if we take lattice-preserving embeddings that project a graph into Rn while preserving the lattice structure, and we can take our axioms, and we can project this using this graph projection entirely into a lattice structure, then we could at least invert this, and we get injectivity, at least. Maybe not totality, but we get injectivity. And the way in which he has done this is by exploiting the category theoretic semantics of the description logic. So category theory, for those of you who are familiar with category theory, you can represent categories in the form of diagrams, and you can give the semantics of logical languages, such as the language that we are dealing with, also as well as categories, and then therefore you represent them as diagrams. So basically you get something like this. An intersection is represented like this, a union is represented like this, in the categorical semantics of the language that we are working with. And then you can also do negation, and it becomes a little bit more complex. And with this, we tested this. So we tested this, and I need to be a little bit fast, but we tested this for the task of entailment. And this is a standard task here where we compared ourselves to a number of other embedding methods for specifically the same ontology. And you can see that there's only one method, and this is this invertible method, that actually can compute entailments in latent space. And this is Ketty. And the gap is huge. And the gap is huge because of the property of injectivity that we preserve in this space. Okay. Let's go back to rare diseases, because this was a small detour. So now rare diseases. What we have been designing is a system that uses these type of embeddings, in which so we call this PhenomNet, or various forms of this PhenomNet. It's a program that we have been working on, or that I have been working on since 2010. And continued working on this throughout my time here at KAUST, and many of my students work in one form or another on this kind of system. And what this does is it's essentially a system that combines two things. It combines some machine learning approach, not ours usually, to detect whether a variant likely affects a gene function, a genetic variant affects gene function, and then it combines different types of background knowledge from different model organisms or from basic biological experiments, and determines whether the phenotypes, the signs and symptoms that we see in this patient could likely have arisen out of a malfunction of a particular gene. So it combines two things. It does a genetic variant, a mutation, affect the function of a gene, and if this function of this gene is disrupted, could it likely lead to these phenotypes? The second part is knowledge-based, the first part is just learning. And in the end, we put this in some kind of, we use formalized theories, so graph-based methods primarily still. This is why CAT-E is relevant for this, where we generate different forms of embeddings, we combine them, so we can combine this on phenotypes, functions, and so on. And then we train this on a very large knowledge graph that integrates this information and combine this, and then we combine this with variant features in the end to train some machine learning model. And in the end, when you look at the performance of this, so this is not a knowledge-based method, and it's not just data-driven, it's a combination of both. But if you combine this, if you compare this, so the first methods are entirely genotype-based, so they only use genetic information. There's also alpha missense and similar methods on this. So this is, there are hundreds of methods that are entirely genotype-based, and they determine whether a variant interrupts a gene function. And the second set of methods, these are the state-of-art methods, which do use the same information as our methods. They use both the phenotype information of the patient and the genotype. So the first one ignores phenotypes. So, and the reason why ignoring the phenotype is not a good idea is that most of us have maybe 15 to 20 genes with a complete loss of function, and we are completely normal. So we need to not just determine whether a variant causes a phenotype, disrupts the function of a gene, but also whether this disruption likely causes the phenotype. And you can see that these are basically now state-of-the-art methods. We have some better methods. They're only available as preprints, so I didn't discuss this, but this is something that my student Asa published last year. And these are now the state-of-the-art methods for variant prioritization if you have both phenotypes and genotype information available. And it's called embed PVP. So we can extend this. So this is very extensible because we can do this also for structural variation. So we have this for structural variation, and I'm not going to go into detail in this architecture because of time. But essentially the same principle can be extended to different types of variants. So there is, so this was mainly for single nucleotide variants. This is for structural variation when large parts of a genome are either deleted or inserted or duplicated, or maybe they're inverted or they're translocated at a different location. So also available as free software for structural variant prioritization based on phenotypes. And all of this is enabled by a set of embedding methods that encode background knowledge from basic biology and from clinical information in a knowledge graph, and then use this together with genetic information to identify which variant causes this disease. And because this is very useful, we have also, because these embedding methods seem to be very useful, we have a library for this. It was, I think, the only library which specifically focuses on description logic embeddings. And we can discuss why this is useful specific for this task, because all our knowledge bases are structured as description logic knowledge bases, but you can get this on GitHub, and we also have this library. So if you want to use some of these methods, so if you want to use them in your own research, and you have structured knowledge bases that you want to use and combine with some data, then have a look at MAUL. It's machine learning with OWL ontologies. It also has a very nice OWL logo. So you can have a look at this. If something does not work, Fernando is very responsive. He sits there. You just raise a GitHub ticket. So you can try this, maybe. Okay. So in summary, we need to have some kind of background knowledge for most tasks in medicine. Rare diseases are in a way the simplest task, And you might think that a common disease, so a common disease like diabetes, so type 2 diabetes, is easier for machine learning models to deal with. And in a way they are, but it depends what you want to do with this, because common diseases are, so rare diseases, these kind of diseases that we deal with are often, almost always monogenic diseases. One gene, one disease, mostly. But if you think about type 2 diabetes, although you have millions instead of hundreds of people, for training, you also have a combinatorial explosion because these diseases are complex diseases. They do not arise from a single gene mutation. They arise from combinations of multiple genetic variants together with lifestyle factors. And for this, even predicting risk is something that is very challenging, and the search space increases. For those of you who are interested in machine learning research, so this was, you have seen, it's basically two parts. Maybe I did not leave anybody satisfied, neither the machine learning researchers nor the people who wanted to learn about diseases, because I tried to put two things together in 20 minutes, which I'm already way beyond. I'm very sorry about this, but essentially this research area is called neurosymbolic AI or neurosymbolic integration, and this is basically the main focus that my talk was about. So if you want to learn more about it or read more about it, search for these keywords. And I need to thank my group. It's already an old picture, so the group has changed a little bit, but most of the people, most of the work I presented is the work of my group, and I want to thank you very much.
So you mentioned utilizing this technique for rare diseases. I want to basically ask, how does this fare first in contrast to using foundation models or things, for instance, like AlphaGenome? And would we benefit from using something like multi-omics modality and perhaps introducing inductive bias, since we're talking about rare diseases specifically? Thank you.
Yeah, thank you. Very good question. So AlphaGenome is based on genotype. It's a model that captures genetic or genomes, and it will allow you to determine whether the function of a gene is disrupted, but it will not help you to determine whether this disruption causes microcephaly, for example, a certain phenotype. Because for this, we need more information. It's beyond genome. It's a different level. What the knowledge graph does here is it sits, so there's a lot of genetic information, and this is where machine learning is really great, and there's very little not that much additional knowledge that we can add. There might be some. So what here the knowledge graph does is it sits on top of the genetic level, on the molecular level, and adds information about physiology, about environmental interactions that we simply do not have on a genetic level. And for multi-omics, I mean, the same method will work with multi-omics, and we also work on multi-omics integration. It's a little bit different because multi-omics integration, of course, we can get much better. We can get much better at pinpointing molecular abnormalities, but the phenotypic consequences of this, and we get a little bit further because we go to protein level, maybe if we go to the protein, but beyond that, to cell-cell communication and so on, it's very difficult still. So multi-omics will bridge this gap a little bit. So far, we are far from capturing physiology or anatomy this way.
Thank you.
Thank you.